Let's dive right into this explainer because today we are looking at something absolutely wild. A monumental, yet, let's be honest, highly controversial breakthrough in human genetics. A recent study actually claims to have rewritten the genetic code of a human embryo, and they use this brand new, ultra precise method to do it. Whenever we hear about editing human embryos, it immediately sparks this massive wave of emotion, right? Awe at the scientific possibilities, but also some deep, profound anxiety about where this is all leading us. So, we're going to break down exactly what this new tech is, what the researchers actually pulled off in the lab, the major biological roadblocks they slammed right into, and the fierce ethical debate it has triggered across the scientific community. But to really get a grip on the stakes of what's happening right now, we have to start with the ghost that kind of haunts this entire field. Maybe you remember this. Back in 2018, Chinese scientist He Jiankui completely shocked the world. He announced he had created the first CRISPR-edited babies. He used a first-generation gene editing technique on human embryos, implanted them, and 2 women actually gave birth. Well, the global reaction was swift and it was furious. Scientists universally condemned the work as just dangerously reckless, mainly because the tech was simply too raw, too risky to be used in actual human beings. The outrage was so intense that He Jiankui ultimately spent 3 years in prison in China for a legal medical practice. That scandal, that is exactly why the scientific community is on extreme high alert whenever human embryo editing is even mentioned today. So let's move into our first section, the scientific upgrade. This new study out of Columbia University proposes a major upgrade to how we edit genes, and it's a massive conceptual shift. This isn't the same tech from 2018 at all. It's a totally different approach. So, the first generation tool, CRISPR-Cas9, acts kind of like molecular scissors. It quite literally cuts right through both strands of the DNA double helix. And because of that blunt cutting mechanism, studies have shown it can accidentally cause the loss of entire chromosomes or trigger this cascade of totally unwanted genetic mutations. Essentially, it's just too aggressive for the super delicate environment of a human embryo. But this new thing, base editing, the second generation tool, is different. Instead of scissors, imagine a pencil with an eraser. It doesn't sever the DNA strands. Instead, it lets scientists erase just a single letter of the genetic code and seamlessly write in a new one. It is vastly more precise, significantly more reliable, and represents a really massive leap forward in our genetic toolkit. Okay, moving on to section 2, the Columbia experiment. Dieter Egli and his team at Columbia University actually used this new genetic pencil on early-stage human embryos. And they didn't just blindly edit. They went after 3 very specific, very important genes. First up is the PCSK9 gene, which regulates our bad cholesterol. By making a single letter edit, literally just changing an A to a G, they effectively switched this gene off. Why does that matter? Well, naturally occurring versions of this switched-off gene are known to dramatically lower the risk of coronary heart disease. Next, they targeted the HBG1 and HBG2 genes. Again, using that base editor to make a tiny A to G change, they mimicked a natural protective mutation that boosts a specific type of fetal hemoglobin. And we already know this protects against devastating blood disorders like sickle cell disease and thalassemia. So they literally went in and rewrote single letters with the goal of eradicating severe genetic diseases. Mind-blowing stuff. But here comes section 3, the reality check. Because honestly, on paper, this sounds like an absolute miracle, right? Just erasing heart disease and sickle cell from an embryo's future. But nature is incredibly difficult to control perfectly, and the reality in the lab turned out to be far more complicated. The most crucial point here is that the team ran straight into a massive biological hurdle known as mosaicism. When we picture gene editing, we kind of imagine this clean, perfect, uniform change across the entire organism. But mosaicism is this phenomena where the edits happen super unevenly. So as the embryo grows and divides, some of its cells successfully get that new A to G edit, while other cells completely miss the memo and just keep the original unedited DNA. You end up with what's basically a patchwork embryo. And think about it. If you're trying to cure a genetic disease, having a patient where only half of their cells are actually cured, that is a very dangerous and unpredictable medical outcome. And unfortunately, the problems didn't even stop at mosaicism. To actually get that base editor into the cells, the team had to introduce really high doses of an mRNA editor. But the sheer dose required, it proved to be toxic. The mRNA caused the embryo cells to just completely stop dividing altogether. It literally arrested the embryo's development.
Dieter Egli himself was incredibly candid about all this, flat out stating that because these base editors can have such damaging effects on the embryo, the technology is, quote, not ready for the clinic. In his own words, he said, in its current form, you can't use it. It's clear as day and night.
Which brings us to our fourth and final section, the ethical clash.
So the science clearly isn't ready for prime time, but just the mere fact that we are taking these conceptual steps, that has ignited a fierce, highly polarizing debate that is totally dividing the scientific community right now. On one side, you have scientists praising this precise base editing as a vital, careful step forward for human medicine. For example, Greg Neely at the University of Sydney argues that compared to that 2018 CRISPR baby scandal, this Columbia study is exactly how science should progress. It's transparent, it's exploring a much safer second-generation tool, and they are openly acknowledging its flaws. For a lot of folks in the medical field, developing the ability to fix disease-causing mutations at the embryonic level is seen as a moral imperative, and this study represents a monumental, positive shift toward that distant goal.
But immediately on the other side of the aisle, critics are sounding a massive alarm. Fyodor Urnov over at UC Berkeley calls this whole embryo editing pursuit a solution in search of a problem. And why is that? Well, because right now, couples who carry genetic diseases can already use standard in vitro fertilization, IVF, combined with genetic screening. They simply screen the embryos in the lab and select one that doesn't carry the disease mutation. So because that safe alternative already exists, Urnov argues that the only real-world application for editing embryos isn't actually preventing disease, it's designer babies. He fears this tech just opens the door to affluent people engineering superior traits into their children, which could fundamentally alter human equality.
And the fear isn't just about some distant sci-fi future of super babies. It's about what might happen like next year. Bioethicist Hank Greeley from Stanford points out a pretty terrifying reality. The barrier to entry here is shockingly low. A rogue clinic could easily look at this exact Columbia University study, see the blueprint, and set up an IVF and genetic testing lab for just a few $1,000,000. Affluent, desperate, or maybe just misguided individuals might jump the gun and actually pay to have their embryos base edited way before the technology is actually safe. And as Greeley bluntly warns, because of the issues like mosaicism and cellular toxicity we just talked about, one result might be really sick kids. The overarching fear here is that the commercialization of this untested tech is going to completely outpace our ability to regulate it.
Which brings us to our final thought for this explainer. The leap from the crude molecular scissors of 2018 to the incredibly precise genetic pencil of today is undeniably a triumph of biology. Yet, as we've clearly seen, mastering the code of life in a petri dish is a far cry from safely rewriting a human being. We are basically left staring at a profound double-edged sword. On one hand, you have the incredible potential to eradicate debilitating diseases from a family's lineage forever. On the other hand, there's the very real threat of rogue clinics, toxic embryos, and a society that could be deeply fractured by genetic haves and have-nots. So, I'll leave you with this to consider. As science inevitably perfects this genetic pencil, are we as a society truly prepared for the era of human baby improvement?